Okay, good morning, everyone. It is my very distinct pleasure today to welcome Dr. Amanda Hummon for our Canary Center Cancer Early Detection Seminar. I first met Amanda, as she just reminded me, over 20 years ago when I was a fresh prospective grad student looking at grad school at the University of Illinois, and she and her now husband took me out to dinner.
I was unsuccessful trying to recruit her to Illinois.
It is true. She did not successfely recruit me.
I tried really hard and I did not, but we became friends. So
we became friends, and over the years, I followed her work very carefully. She does some beautiful work in cancer proteomics. By way of background, Amanda received her undergrad degree at Cornell. She did her PhD at the University of Illinois with Dr. Jonathan Swedler. From there, she moved to the NCI, where she did a postdoc and learned a lot about cancer. She started her career as the independent faculty at Notre Dame and then defected to Ohio State, where her lab is located in the Cancer Center. So we're really pleased to have her today, and welcome, Amanda.
All right, well, thank you so much. Yes, and so Sharon and I have known each other a long time, and it's been a lot of fun watching her trajectory as she's moved through all the various different stages and talking back and forth about our science. So this is my first time visiting Stanford. It was a lot of fun. So thank you so much for hosting me. I've learned a lot about some great science going on your campus, various different groups. And what I've heard about is so many different aspects and going after all these important questions with various different approaches. And so I always like to start my slides with pointing out that as biomedical researchers, we have a real choice of where we're going to site our study. It may not be something that you consciously think about it when you're doing your experimental design, but there's really a large range that we have as we design any given study. So I think the public probably thinks that we do a lot of our work with human patients, with clinical type samples, but in reality, the vast majority of biomedical research can often be done with simpler systems to start addressing some of the basic questions that we can then move into more complicated model systems. And so I want to just briefly look at three of these model systems in the center. And usually I spend a lot of time talking through all the different aspects of these. I think given how savvy this audience is, I probably don't have to go too much into all the various different details. But each of these three model systems has advantages and disadvantages. So probably a lot of people in the room, I know I've done a ton of 2D cell culture. Yeah, okay. So a lot of people have done 2D cell culture. That's because it's relatively inexpensive, it's versatile, it's fast, it's high throughput, right? You can do 15, 30, 6-fold plates. You can screen many, many compounds, but there's some real disadvantages. You know, I don't know about you, and if this is, in my body, the cells in my body are not grown on plastic. If yours are, that you maybe should keep that to yourself, but but the cells in my body are surrounded by other cells. You got complicated chemical crosstalk. That's really hard to recapitulate on plastic, right? So of course we have animal models, and I just want to clarify that there are some studies that have to be done in animal models. I'm not advocating getting rid of animals, but animals are obviously a lot more expensive, and there have been real calls for us to reduce, reuse, and refine our dependence on animal models. The Center for Drug Evaluation and Research at the FDA put out another large statement in June of last year advocating that researchers use fewer animal models and instead start embracing more of the more complicated in vitro model systems that are available to us. So organ on a chip, organoids, spheroids, many of these 3D cell cultures. So with these model systems, they again don't have the full complexity of an animal, but with these model systems, you do have cells that are surrounded by other cells, and so you get some of that complicated chemical crosstalk. So that's the space my lab's been operating in for the last 12 years. We are a colon cancer lab, and so we grow colon cancer spheroids. These were first developed by Robert Sutherland at the University of Rochester. He published an elegant series of manuscripts throughout the 70s and the 80s, where he first described these. He also referred to them for the first time as spheroids. And the really beautiful thing about the spheroid model system is that while it looks like a simple ball of cells, you actually have quite a bit more going on beneath the surface. So when you grow a spheroid, it grows from a central nexus of cells, and the cells divide, and it grows outward in a radially symmetric manner. What happens is that as the spheroid grows to its full mature size, and they grow to be about 1 millimeter in diameter, so it's really not that large, you get molecular diffusion gradients. So if you consider, for example, this little wedge here for oxygen, we have high levels of oxygen on the outside. That's normoxic conditions, normal oxygen. But as oxygen has to penetrate inside and it's being consumed by cells, you end up having lower levels of oxygen in the center. So the very center of a spheroid is hypoxic, almost no oxygen. By contrast, mammalian cells put out waste products like lactic acid. Lactic acid is going to drive down the pH. So the center of a spheroid has a pH at 5.5, which is considerably lower than the external pH of normal physiological 7. So the main thing to remember with the spheroid is that while it looks simple, you do have these radially symmetric chemical gradients, which, as we will see, will play out as pathophysiological gradients as well. So we've been growing these for about 12 years. This is my very first grad student, Hummon, and we modified this Nature Protocols paper from Leonie Kutschitsgaard's lab. And growing the spheroids, we can grow them in 96 or 384 well plates. We typically do 96 just because it's an easy protocol. This is how you can tell I was trained as an analytical chemist, because I really love to slap things on coins to show you perspective. So this is how big a spheroid is. Okay. The beautiful thing about the spheroid model system, again, it's relatively simple. It has this inherent complexity, but it is also relatively simple and it's extraordinarily reproducible, which as an analytical chemist, I love that because we can do method development and we know our system is going to look the same every time. So here's time and culture versus overall spheroid diameter. Every data point here is representing 5 measurements of 6 spheroids, and you can see the error bars here are very small, less than 5% standard deviation differences across the diameter. And I've had about 30 students work on this project over the last few years. And again, the patterns are extremely reproducible, which is really nice when you're trying to do method development work. The other thing about the spheroids, as I mentioned, we have these radially symmetric chemical gradients. That's going to affect the health and viability of the cells. This is flow cytometry data. This is HCT116, one of our colon cancer cell lines. If you take just monolayer cells, so HCT116, grow them in a flat plane, do flow cytometry on them, they're 97% viable, which is crazy. But when you start growing them into 3D, you can see this changes. And we usually do our experiments around day 12, day 13. You can see we have a nice blend of about 40% viable, 60% some flavor of apoptosis, which is actually similar to what the clinical makeup of an actual colon tumor is. So again, with spheroids, if you remember nothing else from my talk today, that the spheroids, because they are radially symmetric, they have these different chemical radial gradients. They also have these physiological gradients. So the outside of the spheroid, we spoke about this morning, growing and proliferating. The center of the spheroid is undergoing apoptosis. Sometimes we'll describe this as the necrotic core. There's kind of we like to say there's a nice mix of death going on in the center. So in the center here, this is staining for trypan blue. You can see the cells are mostly dying. Not all of them, though. A few are robust enough to continue to live. Those are actually the ones that are most interesting because those are ones growing at pH of 5 with no oxygen. So that's a very interesting problem right there. And then at the outside, you can see staining here for proliferation at the very outside, right? So growing on the outside, then a layer of quiescence, necrosis in the center. So we have been using these, we and many other groups have been using these. As I said, they're a wonderful model system because they are so reproducible. We have been using these to look at drug penetration, right? Because when you think about different chemotherapeutics that are developed to treat a tumor, we often think of cancer as having high rates of angiogenesis, development of all these blood vessels. But in reality, if a tumor is truly fast growing and with fast proliferation, the rate of proliferation can be 10,000 times that of the rate of angiogenesis. That is, the tumor is dividing and growing much more rapidly than this blood supply is coming in. So when you think of this from the perspective of someone who's developing new drugs and new compounds, you really want to develop drugs that are going to be able to move from the blood vessel, because most chemotherapeutics are administered intravenously, and you want it to move through the cells and treat all the cells that are far away from the blood vessel. So that's from the drug developer side of things. As an analytical chemist, I, of course, look at this and say right away, okay, we also need to have appropriate strategies to be able to image and look at this overall drug penetration. And so this is the space that my lab's been operating in for the last 12 years or so. We have been doing imaging mass spectrometry, specifically MALDI imaging mass spectrometry. I know there's a lot of expertise in the room to use this type of technique. And so with MALDI imaging, we take a solid sample, in this case, cartoon of a brain here. We take a very thin slice of that brain. We put it down on a conductive surface, and then we spray an explosive over the top of it. That's our moldy matrix. And then we raster the laser. And at each point where the laser interrogates, there's going to be a desorption and ionization event. And what we can do is we can then go ahead and map out those various different species. And of course, the real power in this technique is that we can do it either in a discovery mode or a targeted mode. And if our species is ionizable and we're able to detect it, we're able to generate these various different spatial maps. So this is the space that my laboratory started working in about 10 years ago. I had a truly exceptional grad student, Dr. Xin Lu. We spoke about her a little bit earlier. Professor Zaire was asking me about things that help your laboratory. This is the student I was mentioning, that keeping her in my lab longer was the single most important thing we could do at the time. And so Dr. Lu, her PhD project was taking three-dimensional cell cultures and pairing them with the exquisite amount of chemical information we can get out of multi-imaging mass spectrometry to look at the penetration and metabolism of different types of drugs. And so we started this with irinotecan. Irinotecan is a topoisomerase 1 inhibitor that's given to stage 3 and stage 4 colorectal cancer patients here in the US. Has an M/Z of our molecular weight of 587. And so we were able to demonstrate that if you take colon cancer spheroids, these are grown with HCT116 cells. And if we take the spheroids, we grow them up to their full maturity and then start treating them with irinotecan. This is at the IC50 of irinotecan, so 20 micromolar. What you're looking at is a time course of penetration of irinotecan across time. So each column represents one section. So there's 7 vertical sections here of the spheroid. So after 2 hours, 6 hours of penetration, so on down the line, or treatment rather. And you can see irinotecan fully penetrates after 24 hours, and we see hotspots of accumulation here at 48 hours. Now, of course, mass spectrometry is one of the wonderful things about it is you don't look at just one analyte at a time. It's multi-channel. We have the ability to see other things as well. Irinotecan is a prodrug, meaning that it is the inactive precursor that is given to the patient. That inactive precursor is metabolized, and it's actually metabolized to form a whole range of different metabolites. The one that we care the most about is this one right here, SN38. So SN38 is an incredibly, it's the cytotoxic form of irinotecan, so it's about 1,000 times more cytotoxic. And so this is produced by, through carboxyesterase. It is actually also inactivated into a gluconoride form. So this is a fairly transient metabolite, but that's really the metabolite that we want to track. And so one of the really nice things with mass spectrometry is that since we can detect so many of these different species, we can then also generate these different M/Z ion maps to be able to compare and contrast the distribution of the different species. So in this particular case, we have one of our HCT116 spheroids. You can see the micrograph right here. This in red at 587, that is irinotecan. So in this particular case, that's what we gave to the spheroid. So it makes sense that it's the most intense. And then we've mapped 3 other metabolites as well. And so here are the 3 metabolites. The one we care about is SN38. This is at 393. You can see it's a fairly small peak because again, it's a transient metabolite, but we can go ahead and map it to see its distribution and compare that back against the prodrug. So in this way, we can look at these various different species. One of the things that we really wanted to do was to look at the species, figure out where they are present, but then also to go in and do this in an even more quantitative and rigorous fashion. So in this particular example, we were able to scan through and see what was present, but we also wanted to go in and quantify. And we certainly can do the quantification by imaging, but we also wanted to start bringing in another technique as well. So I want to briefly introduce this technique called serial trypsinization. So I'll talk about this more as I go on through. So serial trypsinization, you can see it going on right here. That's 96 spheroids in a petri dish, just on a rotary shaker. And serial trypsinization, it is another technique that was also developed by Robert Sutherland back in the 70s. So he published in 1971 an article on serial trypsinization. And what this is that you are able to peel off the concentric layers of a 3-dimensional cell culture with incubation with trypsin. So it's a really clever idea. So again, the overall geometry of the spheroid is that it is completely radially symmetric. So for those of you, anybody who loves to cook, right? Okay, so it's like an onion, right? So when you cut open an onion, you see all the layers. Same thing here, okay? So the center of the spheroid, those are those original cells that are necrotic, that are dying out. That's still in the center. And then you have these concentric shells of cells. Okay, this is a slice through it. So you're only seeing it as rings, but it's actually full shells, just like what you'd see in an onion. So we think of this technique as the peeling of the onion. So what happens is that if we pipette in low concentration doses of trypsin, so 0.05% trypsin for 3 minutes, we do that rotary shaking that you just saw. We then pipette out the solution. We can harvest off these various different fractions of cells. And again, the reason this matters is because across these different areas of the spheroid, because of the radial geometry, you have different chemical microenvironments that are present for all these different cells. So the cells on the outside are experiencing, high oxygen, high nutrients. The cells in the center are in this nasty acidic environment. And we can now harvest all these different fractions and be able to do follow-up experiments. Okay, so this was in case my video didn't work, quite frankly. So these are pictures of what it looks like. And here you can see we can collect all these various cellular populations. So to bring this back to our study of irinotecan, so again, we had treated the spheroids with irinotecan and performed imaging mass spectrometry. We were able to see the distribution of irinotecan and various different metabolites as well. And what we wanted to do again was to go in and do a more quantitative study to look at the relative amounts of irinotecan and the different metabolites in the different parts of the spheroid. And we wanted to try out the serial trypsinization procedure because we found the Sutherland manuscript. We really wanted to try this. So Shin repeated the exact experiment. Again, she took irinotecan, treated spheroids, and then she performed the peeling of the onion, pulling off the layers into these various different populations, and then quantifying by tandem LCMS-MS. So doing it this way, you need to add an internal standard if we're going to be quantitative about it. So we use camptothecin. Camptothecin is the synthetic precursor to irinotecan. You can see it looks very similar here in the chromatogram. And as I said, Shin repeated the exact same experimental conditions. So HCT116 spheroids, same dosage, same time course, performed serial trypsinization. She was really heroic. She collected 10 layers of cells. And what we can do then, because we have this internal standard, is that we can go ahead and quantify the amounts that are present of the prodrug. So this is quantification of irinotecan itself. We also purchased a standard for SN38. So we were able to quantify the amounts of SN38, again, in those different layers of the spheroid and across this time course. And so at this point, with the imaging mass spec, we have the ability to kind of scan through, do a discovery experiment, look to see what's present. And then with this more rigorous LCMS-MS, this obviously takes a bit longer, just, full disclosure, but you get very nice quantitative information out of it. Okay, so we had been doing this at this point for about 5 years or so, and I was starting to get a little antsy about trying to make this a little bit more clinically relevant. While it was absolutely fine to start off with taking the spheroids, dosing them in a 96-well plate, I wanted to see if we could make this a little bit more. I'm always trying to move things a little bit closer to the clinic, if possible, with our various different situations. And so, obviously there's a lot, it's much more complicated when you start looking at the human body, absorption, distribution, metabolism, elimination. It's hard to, capture AD and ME when you're dealing with a 96-well plate. Okay. And it was about this time I was at PitCon and I ran into Dana Spence in the hallways of PitCon. And he said, Amanda, I shouldn't say this to you, but I just reviewed this paper you have on treating spheroids with irinotecan and imaging mass spec, and I think I've got a great system that we could use to look at this. And so he has a 3D printed device. I once called it microfluidic, and I was corrected. It's not micro, it's too big. But this is a fluidic dosing device, again, 3D printed. It has HPLC inlets and outlets on either side of it. And it has channels that are running through it. So you can't really see too well here, but there are going to be 6 channels. Each of these channels run through it, run through the side of the device here. And then there are transmembrane inserts that pop into the device. These transmembrane inserts, we'll see in just a second, are porous at the bottom. And what this allows you to do is treat whatever's in the transwell insert with a solution that is flowing through the channel. And the Spence lab, they're experts in fluid dynamics and all things of that nature. And so they had figured out the exact dynamics to be able to simulate very accurately both oral and IV PK curves. So the overall concept, they had been doing this with 2D cell cultures, but what we really wanted to do was put a spheroid in here. So this is just a very crude cartoon here. So you put a spheroid into the transwell insert. So here's the transwell insert again. Again, it has a 0.3 micron pore size at the bottom. So 0.3 micron, obviously big enough to hold in the cell, big enough to hold the spheroid in place, but small molecules can diffuse up into the transwell insert and treat whatever's inside. So if you have a syringe that is filled with drug, right? So the channel here then fills with drug. It's going to flow up into the transwell insert. And again, they have the fluid dynamics worked out. So this is an accurate simulation of the first half of an IV PK curve. If you then swap out your syringe and you just have clear buffer, or you just have buffer, you clear out the channel, which clears out the transwell insert and generates the second half here of the PK curve as well. So what we wanted to do was put spheroids into the device and see if we could accurately mimic a chemotherapeutic regimen. Okay, this is what it looks like from the top. So again, you can see there are 6 different channels. And I wanted to put this in here just very briefly to point out that one of the other really nice advantages of doing 3-dimensional cell culture systems is the sheer number of biological replicates. I have collaborators do a lot of mouse work, and they really agonize over the number of mice that you can afford. With the spheroids here, we put 5 into each well, but you can see we could easily put more. So you have a lot more possibility to do different biological replicates. So you could do different drug doses if you want, or as I'll show you later, treat them all with the same thing and then take them off for different types of analyses. So the first thing that we did was simply go ahead, before we went into the whole chemotherapeutics regimen, we first wanted to make sure that we actually could treat the spheroids with 1 drug at a time. Since we'd done all our preliminary work with irinotecan, we started off with irinotecan as well. This is work done by Dr. Gabe Labonia, one of my grad students at the time. And so Gabe took irinotecan and treated spheroids in the fluidic device. And you see the patterns are relatively the same, that we have the prodrug in the center and SN38 on the outside. So with this fluidic device, we are able to simulate the same process. So then what we wanted to do again is start to simulate an actual chemotherapeutic regimen. And so in this country, for patients who are diagnosed with colorectal, stage 3 or stage 4 colorectal cancer, the 2 most common chemotherapeutics are both combination chemotherapeutics. So you have FOLFOX or FOLFIRI. FOLFOX stands for folinic acid, which is an adjuvant for bifluorouracil. And then you also have oxaliplatin. FOLFIRI is also folinic acid and fluorouracil, but adding irinotecan. They also now give FOLFOXIRI as another popular one as well. But the main thing to know about it is that it's not just a single injection of 1 drug, it's a much more complicated process. So this is an example regimen that might be given to a patient on one of these treatment regimes. So for example, the patient comes into the clinic, they are given folinic acid for 2 hours with either oxaliplatin or irinotecan, given a bolus of 5-FU, allowed to go home, but still getting 5-FU through an ambulatory pump for the next 22 hours. Come back to the hospital the next morning for another dose of folinic acid for 2 hours, more 5-FU, and then another 22 hours of infusion by way of the ambulatory pump with 5-FU. So it's a 48-hour window of treatment with these various different drugs given in different time courses. So again, just not a single dose at a time. So this is one regimen that is quite common. We decided to make things as easy for us as possible while still simulating this. And so we found this is the simplest version of the FOLFIRI regimen. So starting with folinic acid, then doing irinotecan, and then adding in 5-FU. So we went ahead, we treated our spheroids again in the device, in our fluidic device, flowing the different doses through the channel, performing imaging mass spectrometry to look at the distribution of the various different drugs. You can see irinotecan shows the same pattern we've seen before. Folinic acid also favors a similar distribution pattern, although interestingly, some of its metabolites only show up in the quiescent region, which we still haven't figured out why. You can see there's more folinic acid because it's given longer. But anyway, we were able to map each of the 3 different drugs and their metabolites, look at the various different distributions. But the other thing that we decided to do at this point, about half of my lab, as Sharon introduced me, she mentioned that I do a lot of cancer proteomics as well. So about half of my lab does imaging mass spec. The other half does proteomics and phosphoproteomics. And it was at this point that it was really nice that my grad students were all talking to each other. And Gabe was doing all this imaging work. And one of my other students in the group, who's now since graduated, Dr. Caitlin Ludwig, Kate is a real expert in proteomics. And she said, well, I would love to see what's changing in the proteome of these spheroids with the treatment. And so since we had so many replicates, again, because we can put so many of these spheroids in here, we decided to take some of them from all the imaging and then use some of them for quantitative proteomics. So what we did is we harvested off some of these spheroids. We used iTrack quantitative labeling, using rather standard approaches here. We were able to quantify about 5,000 unique proteins, looking at both the 24 and the 48 hour window, seeing 171 proteins, those same ones, but some more at 48 hours of dosing. And what was very nice to see was that a lot of the pathways that we expected to see changed. This is a well-established regimen that we are able to see these. So for example, huge changes in histone protein abundances were detected, and other synthetic pathways as well. So this was kind of a nice proof of concept that for the first time we could really pair and marry our imaging mass spec data with our imaging mass spec data with our proteomics as well. So I'll just briefly mention, I don't want to, I'm sure you guys don't want to be here all day, but I'll just briefly mention that over the last few years, my group has done a number of other different types of compounds as well. So we've worked on platinum species. We've spent a lot of time actually looking at liposomal configurations, how that changes the drug penetration kinetics, and looked at that. And also started looking at antibodies, which I will hopefully get to a little bit at the end, looking at these both individually and in combinations. But this study that I just described, looking at the iTrack and looking at both the imaging and the proteomics, really got me thinking. And this was actually right around the time that I moved my laboratory. So as Sharon mentioned, I started my lab at the University of Notre Dame, and 8 years in my faculty career, I was recruited to Ohio State, which was kind of a nice time to kind of reinvent myself a little bit, right? You kind of, it's a moment of reflection where you think, what do I want to do with my career? And when I thought about what we had done so far, I was very proud of our work that we had done in an area that I would broadly define as pharmacokinetics. So pharmacokinetics is defined as looking at what happens to a drug when it's added to a patient, right? That's the definition of pharmacokinetics that most people would think of. In my lab, the patient is always going to be a spheroid. And so pharmacokinetics, I would define in our lab, is what happens to the drug when we add it to a spheroid, right? But I would say that we were now starting to get into the area of pharmacodynamics. So pharmacodynamics is what happens to the patient with the drug. So in this case, what happens to the spheroid when we add the drug? And we had started doing this a little bit with the iTrack study, and I was very proud of the iTrack study. But at the same time, the one thing that really bothered me about the study, if I'm going to be honest and self-criticize my own work here, is that we homogenized the whole spheroid when we did the proteomics. And that was frustrating in a way because we have these wonderful different layers, and I really wanted to take advantage of the beauty of the model system. So we started thinking about how can we develop a way to go after those different changes in the proteome in those different chemical microenvironments. And so in order to do this, we decided to use isotopic labeling. So SILAC, or stable isotope labeling of amino acids in cell culture, is a very well-established technique developed in the T.S. Mann's lab over 20 years ago now. It's a metabolic labeling strategy, meaning that you add the isotopic labels through the diet. So human cells, humans cannot make lysine or arginine, those two amino acids. We have to take them in our diet. So if you add then either heavy or light isotopes, so carbon-13, nitrogen-15, you can use that to discriminate where the cell came from, right? So if it has heavy label only in the one sample and then you mix them together, because at the end of the day, a mass spectrometer is a big, expensive weighing machine. Let's be honest here, we can discriminate these differences. So in the last few years, people have been really clever using SILAC in new and exciting ways. And so one of the main ways it's being used now is in temporal proteomics. And so this is, I would say, using the same approach that the biochemists have used for many years with radio labels. You add a pulse of a radio label and then you track it across time. Same concept here, that if you add a pulse of a heavy label, you can then track the changes across time. And so this is a figure out of one of Bernard Custer's papers, and he exhaustively characterized a really elegant manuscript. He looked at the changes in the heliproteome across time with pulsed SILAC. But every example that I had seen was using this in a temporal fashion. And what I really wanted to do was see if we could use this in a spatial fashion so that we could use this to complement our imaging studies. So the overall concept here, we call this spatial SILAC, is that can we use this radial symmetry? So I spent the entire first 10 minutes of my talk talking about spheroids and how spheroids are radially symmetric balls of cells. Can we use that to our advantage to add different isotopic labels over time? And I should say this is a project that was developed, one of my former grad students, Dr. Jess Lukowski, she now runs the mass spec facility at WashU in St. Louis. Jess helped me develop this idea. And then Nicole Beller, who is a current grad student, but defending on Monday when I get back, Nicole has really taken this project and run with it. So again, the overall concept is that since the spheroid has this very incredibly reproducible pattern where you have a central nexus of cells and it grows outward in this radially symmetric manner, can we introduce isotopes in the growing pattern so that they only end up being in certain parts of the spheroid? So that when you get the different cells and you detect the different isotopes, you can then infer back where that individual protein came from. Okay. So in order to do this, we needed to figure out our SILAC media. I called up Ben Garcia because he does a lot of multiplex SILAC. His advice to me, stay away from deuterium at all costs. Carbon-13, nitrogen-15 will be your friends. So on his advice, although I remember he warned me at the time that media is really confusing in lab meeting, and he was correct about that assertion. So we now have light media, we have medium media, and we have heavy media. Again, we're adding in lysine and arginine, pulsing them in these different areas of the spheroid. And again, we have 3 classic areas of the spheroid, so this 3-plex labeling works relatively well. So again, the overall concept here, I like to say that we are developing isotopic zip codes. So just like if you get what is the zip code for Stanford? 94305. So if I get a letter from 94305, I'm going to say, hey, that's Stanford University, right? Okay, there you go. So if right here you detect, carbon-13, nitrogen-14, you would know that that's the central layer of cells. So again, just using the isotopic signature to be able to infer back whether that cell came from the central acidic core or the outer area. So Sharon mentioned in my introduction, I am married to another bioanalytical chemist who also attended the University of Illinois, worked with Andy Gorth, Raman spectroscopist. Anyway, and so whenever I have ideas that are crazy, I bring them home in the evening, and that evening I was all excited and I described this to Zach and I said, I think this is a great idea. And he said, Amanda, I think it's a really great idea too, but how do you know that those isotopes are going to stay put and they're not going to bleed, right, all over the place? And I said, well, all right, that's our first experiment, right? That's the first thing we have to test. Yeah. So we went ahead and tested that. So we took heavy labeled cells. So these are fully incorporated, heavy labeled cells, carbon-13, nitrogen-15 for both the lysine and the arginine, grew up the spheroid. So we've got that central nexus of cells. We then allow the spheroid to keep on growing. We swap out with light media on day 4 and grow up the spheroid. We then perform the serial trypsinization. Again, that's the peeling of the onion. So we're harvesting off all the individual layers, and then we do bottom-up proteomics, and we look to see where do we detect heavy labels. And we do see some bleed out. Zach was right about that, but over 90% remains in the core. So we're now at a point where we can develop a probability distribution that when we see a specific isotope, we can link it back to a specific point or a specific area of our 3-dimensional cell culture. So the overall concept here, since again, they grow from the outside in, we swap in different isotopic labels. We can grow it. So it's like an onion, again, growing from the inside out. We can perform serial trypsinization, harvest, and look at cells by bottom-up proteomics. We actually planned from the very beginning that at a certain point, once we had this established, we could get rid of the serial trypsinization, just feed the whole thing into the mass spectrometer and let it, well, homogenize it first. I don't want to clog my instrument. But we're now actually at a point where we don't even do this step anymore. We now know all the probabilities, so we can simply harvest it and feed it right into the mass spec. And again, the isotopic signature should be indicative of where the cell came from. And so therefore, the proteomic response can be correlated back to the chemical microenvironment. So in order to prove this, we had to do a lot of different experiments. I apologize if anybody's colorblind. We need to remake these not in red and green. So these are looking at duplex spheroids. So what we did first was just pulse in one single heavy label into each of the 3 positions and then all the controls as well. So again, what we're trying to figure out is, can we swap out the labels in all the various different reverse labeling permutations, and do we see differences? The ending message is that no, the biology, we do not see differences based on the label. The biology of the spheroid is predominant. You can see here's the PCA analysis, and things cluster and separate really beautifully. This is for duplex spheroids, just putting in one heavy label in each position. We then went ahead, well, Nicole did it. It was a really heroic experiment. This took her about a year to do, with all the various different 3 labels in all the positions, every possible permutation. And when we do that, the data is not quite as clean, it's not quite as beautiful, but it still clusters exactly. So the biology of the spheroid is predominant. We can swap out all the different labels, and the overall label is, or pardon me, the overall biology is what determines the proteomic differences. So now that we have this developed, what we really wanted to do, I started my talk by talking about the FDA, and the FDA is advocating that more researchers embrace more complicated 3-dimensional cell cultures for drug testing. We think this is a great platform for drug testing because, again, we can evaluate how is a cell in an acidic, nasty center of a tumor going to respond to a therapeutic as compared to on the outside where there's plenty of oxygen and plenty of nutrients. And so we wanted to use this Spatial SILAC platform to be able to look at how the different populations within the cell culture would respond to these different therapeutics. So we decided to start with the drug regorafenib. Regorafenib is shown right here. If you watch TV late at night, you might have seen this one it's one of these terrible advertisements where they list all the possible side effects. So this one is known as Stivarga. Stivarga is given, it's kind of the last resort for patients with metastatic colorectal cancer. So if both FOLFIRI and FOLFOX don't work, this would be the next one to try. And this is a multiple kinase inhibitor. So we knew that it would affect phosphorylation patterns, and we anticipated that it would cause some pretty pronounced changes. So here it is, 482 M/Z. Okay, so the first thing we needed to do was establish, first of all, whether or not it actually would move through the spheroid. I haven't shown you any failures. I really should do this in a talk, but we do see some drugs that actually don't penetrate terribly well. And so the first thing we wanted to look at was to make sure that regorafenib actually will penetrate through the spheroid mass as we think it will. So grew up spheroids we tried all the different permutations, again, both heavy and light again, and then dosed them with the IC50, perform both mass spectrometry imaging, and then we also complemented it with some phenotypic assays looking at caspase 3 and 7 activation, so looking at apoptosis. And so this is just to show that regorafenib does indeed move through the spheroid. You can see it takes a little bit longer than what we saw with irinotecan. So this is just showing the penetration of regorafenib into the center of the spheroid over time. And then we do also see activation of apoptosis. So once we established that it is in fact making it through all the cells that we're interested in, we went ahead and performed the proteomics. And so with the proteomics, we have, again, lots of different conditions, and the normalization on this, I won't go into all the details, but SILAC is a lot of ratios of ratios is what I've learned. So, and then analyzing everything on a fusion. And so let's start first by just looking at the whole spheroid. So this is what happens if you take the whole spheroid and you just analyze it, you treat it with the drug, and then you analyze the whole thing all at once. This is what it looks like. And so, you can see huge differentiation here between control and treated. And we see many different proteins that are deregulated. And we have I actually really generally do not like these hairball graphs. I find them not the least bit helpful when people show them in talks, but I'm going to show you one here, and it'll be clear in just a minute why. So this is what it looks like when we treat the entire spheroid. But then what we did is we teased apart the different isotopic patterns. And when you look at what happens if you look at the proteomic response of the cells, again, on the outside of the spheroid, so those are the ones where there's plenty of nutrients, plenty of oxygen, they're happy and proliferating, versus the cells in the core. So again, those are the ones that are existing, pH 5, we're at 5.5, no oxygen. You can see there's a vastly different response. And so I'm showing again 2 of these hairballs. This is the exact same pathway in Ingenuity pathway analysis. And what you can see is that here in the core, it's activated, but in the outer, it is inhibited. And so we see very different responses across this pathway. You can see the volcano plots look very different as well. We see this actually across a number of different other pathways as well, where these are just a number of different cancer-associated pathways. And what we are seeing is that when you tease apart these cellular populations, the cells in the very center, no oxygen, no nutrients, et cetera, they look very different than the ones on the outside. So it really speaks to that if you want this full picture for drug testing, it's worth teasing these different populations apart. So what are we doing with this platform? Now that we have this up and operational, where we can look at, as I said, both the imaging mass spec as well as now looking at the localized changes in the proteome. What I really am excited about using this for is not so much to test drugs that have already received FDA approval. What I really want to do is to use this to be able to test new compounds that are being developed. And one of the nice things about my move to Ohio State is I have several colleagues who are very interested in drug development. So two that I'm collaborating with are shown on this particular slide. So Professors Dewa Pei and Claudia Turo, who are both in the Department of Chemistry with me. Talk about Dewa's compounds in just a second. With the Turo lab, she's developing Rubipi photodynamic therapies, which is a really interesting challenge because that's both drug penetration and then light irradiation to release the drug payload. Really, really fun and cool stuff. But let me just show you a little bit about the work that we're doing with Dewa's lab. So Dewa is developing, this is a whole new class of drugs. Seeing him give a talk on this, he'll talk about how they're changing the landscape of druggable targets, because 80% of the human proteome is not druggable, and their aim is to vastly expand the number that will be druggable. And so with cell-penetrating peptides, you can see this is highly cationic. You can see it is circular. And these particular compounds are designed to break up protein-protein interactions. So if you have protein-protein complexes that have developed, these particular compounds, the cell-permeable peptides, will make their way through the cell membrane, hence the name cell-permeable, and then break up these protein-protein interactions. When Dewa and I first talked about this, I was very excited when he showed me the structure. It's extremely cationic. They have molecular weights between 700 and 2,000, which for a mass spectrometrist, right, that's kind of perfect, right? Okay. And you can see lots of arginine, so really cationic, looked like it would ionize rather beautifully. So Dewa, they had been characterizing these for a long time using different types of fluorophores. So for example, here is one of their compounds right here. Again, they need it to be in the cyclized form for it to work. So here they've attached FITC. And so this is the cyclized form. Here it is opened up into the linear structure. Okay? And so the way that they characterize it, and they still do this to this day, is that they attach a fluorophore and then they use two-dimensional or 2D cell cultures, and they're able to use the fluorophore to look at subcellular resolution. And obviously this has really exquisite, you can see individual organelles, so the spatial resolution here is fabulous. The one disadvantage, well, there are several disadvantages, is that while you can see exactly where it goes in the cell, there's a lot of questions that you really can't answer with this approach. So for example, the first and big one is shown on the slide. If it needs to be cyclized to be active, if it opens up into the linear form, you can't discriminate that with just the simple fluorescence microscopy. But with mass spectrometry, you have the addition of protons, we can easily distinguish that. And in fact, with mass spectrometry, we can look at what different metabolites are formed and other different questions like that. And so Dewa and I have been working together the last 5 years or so. It's really been a very productive collaboration. We've designed an iterative feedback loop between our two labs. So they synthesize the drug, they give it to my grad students. I think this actually bothers my grad students a little bit. They give it to the grad students with a list of masses, and they don't tell them which are the good or the bad. And my grad students go through, they do the imaging, they see where the various different compounds are, and then they report back what they saw. So this is a case where they gave us the compound, and you can see it penetrates beautifully, actually after 4 hours, which is impressive. And they were really pleased because this is the cyclic mass, not the linear, so that was good. But then they were not so happy because this is the primary metabolite. You can see it co-localizes exactly, and that was not a metabolite they wanted to see. So this was a case where we gave it back to them, they altered the synthesis, we then tried again. And as I said, it's really neat because we have this iterative feedback loop where we can use the imaging mass spectrometry to help them guide and target their synthesis. And then of course, the other thing that we can do the imaging mass spec for them. We can tell them where their drug goes. We can tell them how it's metabolized. And now we're starting to also build in the spatial SILAC, so we can also tell them what changes in the proteome. And this has actually been very educational for me because going into this project, I naively assumed that he would be interested in the proteins that he's targeting with his drugs, and that's not what he wants to know about. He wants to know about the off-target effects. What is my drug doing that I don't know about? And that's what he wants me to answer, or that's what he wants us to answer for him proteomically. So it's again, just been a really fun collaboration to be able to do that. So, okay, so the last thing I probably have 3 minutes or so, something like that. Okay, the last thing I'll just briefly talk about is organoids. And so organoids are very similar to spheroids. So spheroids grown from immortalized cell cultures, organoids grown either from primary patient biopsies, or they can also be obtained from mice. So these are not immortalized cultures. And there's real promise to be able to use organoids for personalized medicine. So if you're a fan of YouTube, Hans Clevers out of the Netherlands has a fabulous series of videos where he really, really like convinces you of the power of the organoid. So this is just H&E of an organoid. This is, these are mouse, I think these are small intestine organoids, looking at lipid distributions by mass spectrometry. And so we have been starting to take a lot of these methodologies that we've developed. So again, we develop everything with the spheroids because the spheroids are unlimited. We can grow as many of them as we want because they're out of cell lines. We develop everything with the spheroids, and then we try to transfer our methods into the more fragile and precious organoids. Okay, and so some of the work that we've done, we have worked. It was a great pleasure working with Shannon Mummenthaler. Oops, I'm not going to fall down here. Working with Shannon Mummenthaler at USC. This is also Heinz Joseph Lentz. He is a surgeon out of in the greater Los Angeles area. And so Shannon's a real expert, as you guys know, at growing organoids. And so she and I met at a football cancer research event that Notre Dame and USC had. And she said, I'm growing organoids. And I said, I'm doing mass spectrometry. And the two of us just really hit it off. So we decided to see, could we look at irinotecan? Well, could we look at drugs and use the technologies we developed in her organoid system? So Dr. Lentz, working with patients in the greater Los Angeles area who were willing to donate samples, stage 3 colon cancer. Shannon grew them into organoids, treated them with irinotecan, and then shipped them to us at Notre Dame. You can see here, looking at penetration of irinotecan over that time window. When we look at the various different metabolites, we can also detect those in the organoids. So this implies that we can, with irinotecan, we knew this was a case that the cells would be able to metabolize them. But theoretically, you could use this to be able to look at whether or not a patient's going to respond to a drug that you didn't know in advance they'd respond to. So this is, again, looking at metabolites of irinotecan. And then we've expanded a little bit more. So Shannon was very interested in immunology, and so she convinced me to try cetuximab. So cetuximab, this is treats EGFR, overexpressed in colorectal cancer. It's really big. I remember having conversations with her saying, protein imaging mass spectrometry is really hard, Shannon. So we ended up going ahead and doing on-tissue reduction, just looking at the light chain. But you can see, we can see the light chain. It's very small, but the signal was there. So that allows us to also do imaging mass spectrometry. We had to swap out from our favorite cell line. Our favorite cell line, HCT116, has a mutation, what is it, the G12D, in EGF, or in that particular cell line. So we swapped out and used instead HT29s and DLD1s. So this is looking at the light chain of cetuximab, and then also looking at some organoid patient samples as well. So again, I think there's real promise moving this forward for the possibilities of personalized medicine and to be able to look at these overall changes. So with that, I think that is, yep, that is everything. So here is my current group. If you've ever been to Ohio State, this picture's taken. There's a floating staircase in the big library. So my students voted this is where this year's picture would be. And let's see, I've mostly a group of grad students. I have one postdoc here, Emily Sakara. Everybody else is a grad student, one undergrad back here. A lot of the work I talked about today, Nicole Beller, as I said, she's graduate, or she's defending on Monday. Gonna really miss her. And so she did a lot of the work, and then a lot of the imaging work done by Emily and Egia. And this project is mostly funded by NIGMS. And thank you so much for listening, and please let me know if you have any questions.
Okay, so if you're one of the many people listening on Zoom and you have a question, please go ahead and type it into the chat or raise your hand, and Ashley is going to call those out. For those in the room, I can pass you a mic if you have a question. Parag.
That was great. Thank you so much.
Yeah, thank you.
So I had just a fundamental organoid question for you. You keep on referring to the organoids as being spherically symmetric. So if you had highly metastatic cells in there with motility, would they continue to be spherically symmetric?
So that is an excellent question. So everything I showed you today is based off of monoclonal, one single cell line at a time grown into the spheroid. We are now starting, so actually two of my students right here, so Ariana and Arbil, they're starting to introduce other cell types. We're starting to try to get at this question of greater complexity, because we absolutely have not addressed any. Thank you for bringing that up. Yeah, but we haven't gotten to the point of.
Well, I can actually think it would be really interesting, because you might actually be able to measure the extent of movement, because you might have things with a particular SILAC signature that moved. And so then when you did your trypsinization, you might see things that their SILAC signature didn't match with where your shell of the trypsinization said they should be.
Yeah. Yeah, and actually we, sorry, I'm thinking ahead and linking this to what we're doing right now. So in Arbil's work, we're adding different fluorophores to them, and then using different labeling strategies to start tracking the individual cells. Yeah, sorry, I'm thinking in real time.
Yeah, no, so there's a related series of dyes, the cell tracker dyes, that allow you to track generationally as well. And so those would be really fun to layer on top of this to see how many generations of replication you got in case you had non-homogeneous replication, which again, in more complex systems, you might expect.
Yeah, no, that's a wonderful idea. And I know also, we've only worked in colon cancer, but pancreatic three-dimensional cell cultures are not spherical. They grow sideways like a rod-like fashion. So obviously it's a completely different process, and I have no idea what's going on.
Very interesting. Thank you.
Thank you. Yeah.
Prima?
Thank you so much, Dr. Hummon. So I'm a PhD student here. So my questions will be probably rudimentary, because this is also a new.
Those are often the best ones.
Thank you. So about the different layers that you have for spheroids, right? And I understand you want to label them from from the core outward. Just wondering, the different thickness of this layer. Yeah. And let's say if I want to label it more from outward to inside, so it's not while it is growing, but let's say it's already a spheroid and I want to label them, is that possible?
Okay, so may I ask let me try to answer your first question. Okay, so your first question was about how do you kind of define the layers?
Yes.
Yeah. So that's actually an area of great debate in the group. We spend a lot of time writing out formulas of spheres and things like this. And one of my PhD students, there's Phil right here. Phil's PhD is actually looking at if we do different layers, if we do 10 versus 7 versus 3, how does that change? What are the different populations? And even trying to calculate, because when you think about a sphere, so the cells on the outside, that's a wider area, but what we have found is that, for example, that only the very outside is proliferating. So it's a narrower region. So figuring out how long, for example, do we have to leave the trypsin on to capture that population? And so that is a very, the answer is yes, we are trying to work on that, and that's really challenging. Yeah. To answer your second question, so you were asking, once we have the spheroid established, can we then label it back in? So I wish we could. So what happens when you grow a spheroid is that it grows up to about the 12 days, and then it stops. I don't know if you guys noticed, but in my growth curve, the growth curve that I showed, they cap out. They don't grow beyond 1 millimeter. And so what happens is they grow to a certain size, and then what happens is they stop, and the relative percentages of the different populations start changing. So what happens is that necrotic core gets bigger and bigger and bigger, and it consumes the entire center. So one of my former postdocs did an experiment where she just grew them as long as possible and did flow cytometry at all these different time points. And what we found is by day 50, if we really let them go, they're really no bigger, but you have this very thin shell of 3 or 4 cells deep of cells that continue to be happy and proliferate, because we kept on giving them media. So they continue to be happy on the very outside, and then the entire center pretty much becomes just death. And it's like that scene in, you seen the movie Ghostbusters?
Yes.
Okay, you know the scene when Ben Beckman's like, you want to keep it in the public library, that gooey gross, that's what the center of the spheroid's like. Yeah, it's just.
Okay. Just a follow-up questions on that in terms of it's not clearly defined, right? The
It's not
boundary.
clearly defined.
So
yeah.
when you label them, like, what is the percentage of bleeding that you see between like the core and then the middle layer and the outer layer?
I don't know those numbers off the top of my head. We have them all calculated.
Uh-huh.
So what we do is we'll grow them 12 days and we'll do 4-day pulses.
Okay.
Yeah, so we do know all those numbers, but I don't know them off the top of my head.
Okay, thank you.
But no, you end up thinking all about what is a sphere. And I mean, it's just, if you ever visit me in my office at Ohio State, I have all these children's toys in my office to demonstrate different properties of a sphere.
Yeah, sorry, because I'm an engineer.
Yeah,
so.
no, yeah.
And so
Thank you.
that
That
was
was a very nice talk.
Thank you.
I have a question about the 3D cell culture in general. So let's say if a drug takes about 72 hours for the uptake, is this the same as a patient is going to take? Like, how does it correlate with the real life
Yeah,
situation?
so this is where our model system is a bit limited. So with a patient, it could be different because they're going to have vasculature. We don't have that. So the questions that we are able to ask is whether or not a drug will penetrate or not necessarily is that going to correlate patient, but that's a fabulous point.
I see. So also regarding the metabolites, like I'm sure there'll be some differences, right? If a drug metabolizes in the body and it generates some metabolites, it will be different than in the
Yeah, the liver is important.
Yeah. Have you done any comparative study? And it will be.
You mean between animal models and
Yeah. Yeah.
So everything we've done has been in the 3D cell cultures, and then
I see.
it can be translated.
I see.
But
Also,
no, you're absolutely right. There's only so much metabolism that's going to go on in the epithelial cell.
yeah. Sorry, just a different question.
Yeah.
In the kinetic study for the drugs, how are you determining the dosage of the drugs in like?
Oh yeah, that's really painful. So we do IC50 curves, and we determine the reason that it's painful is that a lot of the assays that are out there are for smaller 3-dimensional cell cultures. This is actually a huge pet peeve of mine. So the instrumental vendors are selling a whole bunch of various different kits, but they're only for very small spheroids. And nothing against very small spheroids, but if it's a very small spheroid, you don't actually have all those gradients. You have to grow them out the larger amount of time. But sorry, to answer your question, yeah, we do IC50 curves, and all the doses I showed you today are at the IC50 doses. And when it's very important to state, the IC50 is for 3D cell culture, because 2D cell culture is completely different values. It's much easier to dose cells in 2D, and they'll die like crazy. It's totally different dynamics in 3D.
Thank you.
But thank you.
So we have some questions on Zoom.
Great
Ashley.
talk.
Oh, thank you.
Go ahead.
Dwayne, go
On
ahead.
Zoom.
Okay, great talk. I have a couple questions. I don't know why it's just echo. Yeah, so I have a couple questions. One, how does protein biologics, so if you were to use this, because it seems like you're looking to study small molecules, and so, or how small molecules affect these 3D cell cultures or the spheroids. So using protein biologics, would that cause a different response, or could you actually see the response that those particular therapeutics cause? And another question, why just colon cancer? Why not glioblastomas or ductal carcinomas or one of the other prevalent cancer types?
Absolutely. Okay. Thank you so much for your questions. So your first question about using protein biologics. So we would be capable of looking at what are the changes in response, what are the metabolic changes. In terms of looking at the proteins themselves, that's a little bit harder. That is doable now by multi-imaging mass spec, but it's more readily accessible for us to be able to look at what are the changes in response to a particular compound. We did show a little bit with the cetuximab. We actually have looked at that data set again, and we have looked at what are the metabolic changes that occur in response to the cetuximab treatment. And that's actually where things get most interesting, because we can see changes as early as 12 hours when we look at what is the metabolic response. So that's, as you correctly stated, looking at the small molecules is much more readily easy to do. And I'm afraid I have now forgotten could you please ask your second question again?
Why just colon cancer?
Why colon cancer?
Ductal carcinoma or glioblastomas?
So my family has a history of colorectal cancer, which is why I have personally chosen to study colorectal cancer. But you are absolutely right that this technique is amenable to many other types of cancers as well. And I think that there's real applicability to expand this this overall process. Yeah, thank you for your question.
So there's another question on Zoom from Ukan Mercy. Thanks for the wonderful talk. How do you see the emerging 3D biofabrication cancer models compare to organoids in terms of studying the questions that you're interested in?
Well, I mean, it depends on, you know, what different, you know, yeah, it depends on the various different questions, right? And so, you know, I think for the things that we've looked at here to be able to assess, you know, drug penetration, et cetera, but getting at some of these more complicated questions of timing and more amenable to those types of questions. It's all about matching your question to your appropriate system. And so the spheroids, yes, they have their limitations, but they also are very good at answering some of these.
Great. So I think in the interest of time, we'll thank Dr. Hummon again for her talk.
Thank you very much, everyone on Zoom.
Yes, and all our friends on Zoom. I think there's like 40 of you. Reminding everyone that next month we have Dr. Brian Cunningham from University of Illinois. In May, we have Manish Kohli from University of Utah coming to speak, and next fall, Olivia Eberlin talking about some of her mass spec imaging work. So thanks again, Amanda.
Thank you very much.